Okay, good morning, everyone. So we're going to get started now. So my name is Mackenzie Woodman. I'm the Geology and Remote Sensing Application Specialist here at Spectral Evolution. And today I'm going to go over a short study that is analyzing soil and clay mineral identification in soils. So first I'll start off by introducing Spectral Evolution. Spectral Evolution is the leading manufacturer in high quality UV VIS NIR technology, field spectrometers and spectroradiometers. We're established in 2004, so we've just celebrated our 20-year anniversary, and we're located in Haverhill, Massachusetts, essentially the Boston area. So our primary application areas and where most of our customers are in hyperspectral applications, remote sensing and vegetation, mining and geology, as well as photonics and calibration transfers. So during this study, we used a host of our different instruments. We used the NiR and the brand new NiR Ultra. So this is the NiR Ultra is the one that is purple, and the NiR is the one that is red. And the Ultra has a ultra high spectral resolution at 1.5 nanometers in the VIS, 3 nanometers in SWIR1, and 3.8 in SWIR2. This is currently the highest resolution field spectroradiometer on the market. And we also have the NiR, which is the second highest spectral resolution for field spectrometers or spectroradiometers, with a 2.7 nanometer in the VIS, 5.5 in SWIR1, and 5.8 in SWIR2. We also compare to the RS-3500 as well briefly in this study. That is to give a representation of a standard resolution spectrometer. So I'll start off by explaining what spectroscopy is. So spectroscopy is the study of the electromagnetic spectrum as emitted or reflected by various materials. The electromagnetic spectrum is a representation of electromagnetic radiation, and they're defined by their wavelength. A reflectance measurement directly relates to the material's chemical structure and bonds within a material. You can think of your eyes like a spectrometer in the way that it can measure wavelengths, and it interprets these as colors, and the wavelength range is the visible light in your eyes. So our spectrometers typically have a range of 350 nanometers to 2500. Spectrometer reads this radiation, and with a radiometric calibration, so that would be the remote sensing series of spectroradiometers, it can also measure the energy across the electromagnetic spectrum, as opposed to just a ratio of detector counts. Reflectance spectroscopy focuses on the radiation reflected off of a material. So you understand the energy before it hits a material, which would be your reference panel, and then you measure after it's reflected off that material, and the ratio would be the effect of that material and characteristic of the material. So what are the applications for spectroscopy in soil science? Typically, reflectance spectroscopy is used in mineral analysis, total organic carbon, total carbon, nutrient analysis, water content, and many more different studies. These metrics are very commonly used in precision agriculture and environmental science applications and research. The idea behind soil spectroscopy is that it can be used to characterize a soil in many different ways with minimum to no manipulation of the samples. Reflectance, it's important to understand that reflectance measurement is going to be a surface measurement. so they do not penetrate the soil. So if you want to get a transect of a soil and scan along a transect, that would be a better way to get a representation of what's underneath the surface. Or you can homogenize different layers within a soil. But here on the right, you can see our NIR as well as our benchtop probe. That would be these pucks you fill with loose materials like soils or clays, and it allows you to take very controlled scans of different soils. This would be if you were taking samples back to the lab instead of taking measurements out in the field. And here on the bottom right, you can see just how reflectance measurements and absorption features can change based on the size of the sediment or just the grain size or other constituents within the soil. So some problems that we're trying to address by using or bring light to is that traditional soil sampling methods are slow and they can be expensive, both with your time and money. There's very few locations in the United States that have accurate soil maps, and so soil mapping is a big initiative. And considering that there's growing concerns about more efficient agricultural practices and effects of climate change, research and over-fertilization with chemicals, erosion, and pollution, it's a very topical issue to try to find a way that you can very quickly analyze soil and create those soil maps. So this study is going to go over different ways of analyzing soil that is fast and efficient, which is soil spectroscopy with a spectroradiometer. But first, I'll start with what is soil. Soil can be described as a mixture of solids, liquids, and gases. The solid portion is made up of organic and inorganic material. An ideal soil mixture could be 45% mineral, 5% organic matter, 25% water, and 25% gas. But of course, many different soils have different constituents. But clay minerals are a major inorganic component and are layer silicates formed from weathering of other silicate minerals. They contain a large percentage of water within the sheets and can hold dissolved plant nutrients. This affects the cation exchange capacity, which is a measure of a soil's ability to hold positively charged ions. And this influences the soil structure stability, nutrient availability, soil pH, and soil reaction to fertilizers. An important point to take away from this is that soils are a mixture, and so they'll be optically complex, which is part of why this study this short research project is important, because it's difficult to tease out the optically complex nature of soils, but it's certainly possible using the technology that we have. So soil reflectance can be broken down like this. Individual molecular bonds vibrate, absorb light with a specific energy quantum related to the difference between two energy levels. Energy quantum is directly related to frequency and inversely related to wavelength. The data can provide a picture of a soil's health, fertility for food source management, land conservation, development, and by understanding what wavelengths and absorption features are dictated by what materials or conditions, it is possible to identify the mineral composition or other things like total carbon or total organic carbon. And many of these metrics are measured through a partial least square regression model that uses known samples to predict these. So when taking a reflectance measurement in the field, it can be conducted with either a contact probe, or for a larger area sample, it can be done with a bare fiber or a lens. So that would be a cone reflectance. So that would be how far you are from the ground would dictate how big your area is. But with a bare fiber lens, you'll have a much broader sample size. So typically, I would suggest the more targeted contact probe measurement. This is in part because the light bulb inside of it has a very high energy, and if you're taking samples in dark soils, this will help you bump up the signal and get less noisy data. It also is less dependent on weather conditions when you're out in the field as well. But for this study, we selected clays that were very common. So we selected kaolinite, halloysite, illite, and montmorillonite. These are selected, well, the kaolinite and the halloysite was specifically selected because they are both within the kaolin group, and they are very similar structural habits. And these different kinds of clay minerals can tell a great deal about the amount of weathering that has taken place, as well as the soil conditions, nutrient availability, etc. So understanding the composition of a soil is hugely important to understanding the environmental conditions and different effects and relationships that are going on within the soil. So, like I said, halloysite and kaolinite are two of the clays that we selected just to give a challenge how similar spectrally these clays are. I wanted to see if we could differentiate them in a mixture to kind of replicate what you would see out in the field. So the reason these are both dioctahedral one-to-one layer silicates and are hydrated aluminum silicates. Halloysite has the same theoretical chemical composition as kaolinite, except for its higher water content, because unit layers in halloysite are separated by a monolayer of water molecules. And you can see that demonstrated on the right here with the water molecules between the two. So important to understand that these spectral reflectances are very dependent on their chemical structure. And so because kaolinite and halloysite are very similar chemically, their reflectance is very similar. However, there is some telling differences between the two that I'll go into more detail later. But to conduct this study, we mixed kaolinite and halloysite into soil at different percentages. The samples were homogenized and scanned, and they were taken at 5, 10, 20, and 50 weight percent in soil. A mixture of 40% soil, 30% kaolinite, and 30% halloysite was also scanned to give an understanding of how, if we can unmix them if they're together, which would be typical. We also scanned illite and montmorillonite at 20% concentrations in soil to see if we could differentiate that as well. And once the scans were taken, we were able to take all of the data collected into EasyID software to unmix the optically complex spectra. And so here you can see the results of just the pure samples, just so you have an idea of what the reflectance signals look like when they're not in a mixture. So in the middle, this one in the middle, the spectra, that is the raw soil with nothing in it. And so as you can see, it's not very optically complex. It does have some features at 1400 and 1900. These are primarily due to water that's in the soil. And then on the bottom two, that's your halloysite and kaolinite, and as you can see, these have very optically similar features, but they are different. And we'll be grabbing or understanding those optical differences and using that to differentiate what is in the soils later. And then you can see montmorillonite and illite both have spectrally unique reflectance. So here you can see the spread of the concentration of halloysite. So the bottom line is the 50%, above that is the 20%, the 10, and the 5. These got shifted up somehow. So these spectra, as you can see, with less concentration in the soil or less concentration in a mixture, these features are going to be less of the reflectance, and so they'll have less influence on the overall curve in your spectra. But as you can see, even at 5%, the light blue line, it still has characteristic features of the halloysite. And so now moving forward, this is the kaolinite concentrations. And as you can see, these features are very different. There's sharper features at this 1400 absorption feature. And even at the 5%, you can still see that doublet right at 1400 that is a characteristic difference between halloysite and kaolinite. And so one of the things I wanted to highlight is the importance of spectral resolution, because with such small differences in features, the spectral resolution becomes very apparent, because the coarser resolution, like the standard resolution 3500, although it can tell the difference between the two minerals, these features start to be averaged out, and you start to lose information here. So primarily on the right here, that's the kaolinite feature, and it actually loses that doublet with the 3500 in the red line compared to the NaturSpec Ultra with the ultra high resolution. You can see that doublet, and the features are much sharper, which is averaged out with the coarser resolution. So these are important things to understand, especially if you're going to be targeting minerals that are very optically similar. It's not that there's still the ability to differentiate these two, but it may take a higher resolution instrument to do it. So here is a breakdown of the halloysite, kaolinite, montmorillonite, and illite, and how they correlate with our spectral library. As you can see, the halloysite and kaolinite with the same match regions correlate very highly, and they correctly identify the mixed clay minerals. Remember, these match regions are adjustable, and I define them specifically due to the features that I knew were present after scanning the pure clay minerals. And you can find these spectra in literature if you'd like to see where the features would be. So in a similar way, I could scan the topsoil with a contact probe in the field and be confident that when it's pulled into EasyID automatically, that it will come with the correct identification. So here are the other two clays. And as you can see, with the defined match regions, they're still correctly identifying the correct clay that is mixed in. So this was 20% weight percent of the clay to the regular soil. So when a spectra is sent to EasyID, which can be done automatically on a scan, it will automatically calculate the derivative and normalize the data for comparison to the spectral library. So you'll get automatic regression or correlation results with the spectral library. And there's several mineral libraries to choose from within EZID. But this kind of takes the legwork of data processing out of the equation by doing it automatically when you take a scan. So it makes for field work very efficient, and it allows you to log what kinds of minerals you're seeing in different areas and do those soil mapping projects. And so as you can see, all of our minerals were highly correlated with the correct mineral. In order to test this a little bit further, I also did a mixture of 30% kaolinite, 30% halloysite, and 40% soil. And as you can see, we're still able to get highly correlated spectra. So it highly correlated with kaolinite, which makes sense since halloysite and kaolinite are very similar. But this is the only time that we had both of them highly correlated, which makes sense because they were both present. So we've identified that both of those clays are present by using, again, the same match regions. So that was a successful identification of different clays, even when they're mixed together. So in conclusion, spectral analysis in soils is a critically useful tool. It is fast and accurate and more efficient than many of the traditional methods. It also has the added benefit of not needing to be, not manipulating the sample or destroying the sample in any way. It can be done in a way that preserves the sample completely. And it can be done in the field and recorded in real time. So this kind of analysis can not only be used for clay mineral detection, but also to measure total carbon, soil organic matter, total nitrogen. It's a great tool to be used in the field, but it can also be used in the lab. So all of the spectra that's collected in the field can also then be further analyzed in the lab, or samples can be collected and taken back to the lab for further analysis or more targeted analysis. If you get into the field and it's a rainy day, it may be beneficial to allow these soil samples time to dry out and bring them back into the lab and scan them that way. There's many different ways that these can be analyzed, but it's inherently non-destructive how we analyze these materials, because it is just a light reflectance measurement. So field soil reflectance spectroscopy is an effective and diverse application for soil science, environmental science, and precision agriculture. It's a very useful tool, and it's a critical tool for clay mineral analysis. So thank you very much, and I'd be happy to answer any questions that you have now. So thank you.